Okay, our next presenter is Dr. Cy Griffin from StemBioSys.
Thank you. Hello, everyone.
Let me just hit play for you.
Great. Being one of the new kids on the block, I'd like to start by thanking the organizers and probably specifically the Alliance for Regenerative Medicine for giving us this opportunity to come before a group like this and talk about our technology and our company. This is actually the first opportunity that we've had to do something like this. So first, just to talk a little bit about our focus. What we'd like to do is we'd like to use these technologies that we have to isolate and expand potent stem cells for clinical use. That's the key goal for StemBioSys at this point. I'd also like to start out being the new kids on the block, telling you a little bit about our company and who we are. The co-founder and inventor of the technology, Dr. Xiaodong Chen, this is actually a technology transfer from the University of Texas Health Science Center at San Antonio. Dr. Chen has a joint appointment there, both in the dental school and in the medical school. His training was as a surgeon at Shanghai. That's where he received his MD. He got his PhD at Nebraska. Did work at Johns Hopkins, and he spent over 20 plus years doing stem cell research at some very nice organizations, Yale, NIH, Arkansas, and the Health Science Center in San Antonio. Co-founder is Dr. Stephen Davis. Dr. Davis is a practicing physician. He's a clinical professor also at the Health Science Center in San Antonio. He's been the principal investigator of well over 100 clinical studies to date. His training was at Yale, UT Southwestern, and UC San Francisco. Myself, Cy Griffin, I'm the Chief Operating Officer. My background and training was in cell biology from Baylor College of Medicine. I've spent most of my career in industry doing research, product development, commercialization, all in regenerative medicine. Most recently spent about 9 years with Kinetic Concepts in San Antonio as our Senior Director for the Center for Advanced Research and Technology, and then also spent 15 years with a company called LifeCell Corporation as their Director for New Product Development. We have a long list of strategic advisors. We're just listing 3 here, but Dr. Davis is pretty well connected, so he's brought together quite a group for us to start out with. Our main advisor at this point is Mr. Gary Frazier. Gary has over 40 years of life science experience, spent a lot of time in pharmaceuticals. He was previously the CEO at OSI Pharmaceutical. While he was there, OSI developed and marketed a cancer drug called Tarceva, which I believe has well over $1 billion in revenue at present. He's also on numerous public and private boards. Kathy Burzik, my previous boss. Kathy has been in bioscience for 30 plus years, development management. She was most recently president and CEO at Kinetic Concepts in San Antonio. She was the president of Applied Biosystems and also has been on numerous boards. So these two have been a lot of help to us. On the development side, Gabi Niederauer also has quite a bit of experience, 25 years in development of biomaterials, primarily in orthopedics and dental, now currently in ENT tissue repair. She's the Senior Vice President for Technology and Development at Intrigue Surgical, previously Director of R&D at Osteobiologics, which was acquired by Smith & Nephew. So let me start talking about the technology. I don't have to tell this group here the number of applications or potential applications for stem cells. In our case, primarily mesenchymal stem cells. And what we've kind of focused in on are a couple of unmet needs. There's a lot of work being done, obviously, from what we've seen today on the ability to expand the numbers of cells. These types of applications are going to take large numbers of cells. So an adequate quantity of these cells, but just as importantly is you need to maintain the purity of those cells and the stemness of those cells. So that's what StemBioSys, the 2 technologies that I'm going to speak to you about, that's primarily where we're targeting. So just very quickly, a highlight of the 2 different platforms that we have. The first one we're currently naming XC Marrow ECM. This is a patented 3D matrix to rapidly isolate and expand stem cells for research and clinical use. when Dr. Chin invented this technology, he took the approach that in order to maintain these cells and get these cells to divide quickly, you needed to recapitulate what these cells see in their normal environment in the tissue. So, you know, this isn't a coating of fibronectin, this isn't a coating of collagen 1. There's over 70 different proteins that are in this extracellular matrix that's been produced. And what he does currently for one of these applications is to start with bone marrow stromal cells, taking these cells, putting them into a dish, allowing these cells with some stimulation to produce their own extracellular matrix in that dish. So this extracellular matrix, once we're done, is about 80 to 100 microns thick. We decellularize it when we're done to get rid of the cells. So what you're left with is this three-dimensional matrix that, again, this is an environment that stem cells are familiar with. So when you come back and use this substrate to seed stem cells, they don't commit. They proliferate very rapidly. Again, they're in an environment that they recognize and they're happy with. And they maintain stemness. And I'll show you some results from some of the work that we've done that validates that. Again, the first one, that's our core technology. That patent was issued in December of this year. The second platform technology came from umbilical cord blood stem cells. And it was utilizing that core technology of the extracellular matrix by which Dr. Chen found a rare subset of stem cells within umbilical cord blood that show pluripotent potential. They're in very low numbers. And these cells share a lot of similarities from a couple of other groups that have described a similar cell set out of umbilical cord blood. They've been shown to be present in anywhere from 4 to 30 cells in a billion in the cord blood. What we found with this is that when you take this cord blood and seed them on this extracellular matrix, the cells have a very high affinity for this extracellular matrix. And in fact, when you take wash cells off of plastic, you've tried to seed them on plastic, you take the wash and you put them on this extracellular matrix, you get a lot of cells that attach. And as you continue those experiments and do the colony-forming efficiency assays or colony-forming unit assays, we see that we're also able to get a lot of cells coming from those wash cells. So these cells don't attach very well. So that was one of the key issues here that allowed us to get beyond the isolation and expansion of this rare subset of cells. But again, it was using that core technology that got us here. So now just a little bit more detail. This is on the first one, the Marrow ECM. You'll see in the left-hand panel, again, comparing to standard two-dimensional cell culture plastic. If you go through a few different passages, up to passage 2, you can see the numbers of cells that we get with the ECM versus standard 2D plastic. And this red line that we put across here is roughly what you would need for a dose. If we look across the board at some of the work that's being done and some of the clinical studies, for an average size person, you're usually looking at 80 to 100 million cells. So by the time you get out to 2 passages, again, starting with just a small vial of, in this case, these were bone marrow mesenchymal stem cells, you can get, very, very quickly, you can get upwards of 3 doses. Whereas using standard methods, you're not even going to have a dose. And the panel in the middle, again, is just to show the economy and the effectiveness of these cells when grown on ECM. These cells, we seeded the same numbers, and now they've been induced to form bone. You can see on the bottom 3 panel, the increased amount of bone that's produced when they're cultured on this extracellular matrix. Also, one of the markers that are constantly used are these cell surface markers. So this is maintaining the stemness or the pluripotential state of these cells. Again, going out, I believe these were done at passage 2. You can see that the ones that were cultured on ECM maintain a higher percentage of cells that are giving you a positive signal. So if you get a lot more cells starting out and more of those cells or a higher percentage of those cells are still expressing these stem cell markers, you get several-fold more potent cells in the same period of time than if you'd used normal cell culture plastic. This is the technology from the umbilical cord blood. Again, it's a pluripotent mesenchymal stem cell. Again, research that Dr. Chen had done after isolating and expanding these cells to some extent. He took these cells and injected them into a nude mouse. You can see the micrographs at the bottom. Sorry. This is from one animal injection and a biopsy from where he had done those injections. You'll notice that there's tissues from all three developmental germ layers that were produced from these cells. So this is a very potent stem cell that he's isolated from the umbilical cord blood. Most people have shown, and we will continue to do studies to validate that these are immune privileged cells. Therefore, you can use them off the shelf. They're not derived from human embryos. It's something that we've tried to stay away from. And we think they're scalable. Again, for the reasons that I mentioned before, we can get several-fold increases when we seed these cells onto that extracellular matrix versus normal cell culture plastic. Just to look at some of the activities and some of our near-term goals as we go forward, we need to further characterize this ECM. This company has been really active probably for just over a year. The company was formed 2 years ago, but it's been more active in the last year. There's still a lot of work that we have to do to further characterize both the ECM and those cord blood stem cells. We'd like to get to the point where in the next 12 to 18 months we initiate proof of concept studies, looking at some of our target applications and working with partners. Any company our size, we're going to be very aggressive about protecting and pursuing additional IP. That's going to add value to our company as we go forward. We're here, obviously, to evaluate the potential for licensing agreements. This, again, as I mentioned, our platform technologies. There's a lot of different potential applications for both of these technologies. Seeking collaborations in the US, China, and elsewhere. And then we're currently in our first round of funding. This is with angel investors. All of the money that we've used to date, or the vast majority of that, has been given to us by Dr. Davis. So we're going through an angel round of financing right now and hope to close that soon. And as soon as we do, we'll be bringing in some additional players to help us get through this. Excuse me. So in summary, we have innovative patented and patent pending technologies, both enabling to current stem cell markets and current stem cell types. There are broad applications for this technology, providing a number of significant opportunities in a bunch of different fields. And we're currently validating and accumulating additional data to position the company to move forward at this point. Thank you.